One. How is everyone doing? I'm going to be your nice guide down the metabolism of glycolysis in citric acid. Doing great. I see that we have some of the same people here, some new people. Excellent. All right, so here's the deal. With the new MCAT, we do actually need to know more of the enzymes that show up and are necessary for our cellular respirations. Okay, this is something that is a new thing on the new MCAT. So we really want to make sure that you're understanding the different steps. There are 3 different metabolism sessions. This one is specifically only going to go through glycolysis and the citric acid cycle. So we're really just looking at the first couple of steps of what we have. Metabolism 2 will go over oxidative phosphorylation. So we're going to really be talking about what you read in your sessions. So as you read these in your chapters, this is going to be a nice, good review for you to really make you sure that you understand. All right. Hopefully we're ready. Hopefully you guys all have some glucose in your system and we're all ready to do this. So in this episode, we've got 2 main goals. The first goal is to identify the function of the key enzymes in glycolysis. And the second one is going to be to identify and describe the steps in the citric acid cycle. Now, this is part of the science knowledge that the MCAT says is necessary for your science skills. All right, in our first section, we're going to take this whole chart and we're going to break it down into smaller, more digestible pieces as we go through. So in this session, we're going to learn to really look at glycolysis. Before we start, I want you to notice that in these first couple of steps, we have ATP being used. So we have ATP input. It's only later on that we get that ATP back out. Okay, it's only in later steps. The first couple of steps are really going to be using ATP. So let's go ahead and go through these first 2 steps, or these first steps. So our very first part of this, where we're using up ATP, we're going to start off with step 1. And this is taking glucose and phosphorylating it to make it glucose 6-phosphate. So in step 1, we all start with glucose. We know what glucose looks like. Okay, glucose is a common molecule. We should know what it is for test day. And what we do is we actually pull a phosphate from ATP. That's where we get that phosphate. And what we're going to do then is phosphorylate the 6th carbon in that glucose molecule. And this is done by a very specific enzyme, and this is a kinase enzyme. When we look at this, this is hexo because it's 6. We're going to be phosphorylating the 6th carbon. So this is our hexokinase. I do want you guys to notice something here. Notice the magnesium. Kinases require divalent cations for their activation. Okay, so notice that all kinases that we see today, we're going to see a divalent cation attached to these guys. Okay, so with our hexokinase here, we are using up an ATP. So ATP is hydrolyzed. Then, here's the deal. This is irreversible. Tell me what that means about the type of reaction. Is it exergonic or endergonic? And tell me about my delta G. Okay, now tell me about my delta G. It's actually negative. It's a large negative. So here's the deal. This is actually exergonic, guys. So this is exergonic because I release energy. Okay, any time that I break a phosphate bond, I'm releasing a bunch of energy. So because energy is released out of the system, it's exergonic. So my product has a much, much lower energy than my substrate. Yeah, so it's overall negative, which means what am I going to have to put a lot of in to go backwards from glucose 6-phosphate to glucose? Yeah, I'm going to have to put a bunch of energy in. Do I have the energy to do that? No, and it's going to be more energy than an ATP, most likely. Yes, Tristan. So when I look at this, that's why this is irreversible. It lets off so much energy that to go back to glucose, it's just not going to happen. Okay, so this has a very, very large negative delta G. Here's another thing to know, is glucose can easily cross the plasma membrane, can easily go into, out of all of my cells. Glucose 6-phosphate cannot. So as soon as I make this as glucose 6-phosphate, it is trapped in my cell. It can no longer leave that cell. Okay? Now, just like any enzymatic reaction, there is some inhibition. And the inhibition that we would have here is if I have a bunch of glucose 6-phosphate, that's going to go back, that's going to have a negative inhibition on my hexokinase and say, whoa, 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 you have way too much going, so you need to slow down. Okay, so as we go through, we're going to talk about what things would activate and what things would deactivate or inhibit many of these enzymes. Okay, so we all know the product of an enzyme reaction, if there's too much of it, can definitely inhibit the enzyme itself. Ah, yes, Nadine, that is negative feedback. And most processes, especially in the endocrine system in our body, are actually negative feedback. Okay, very, very rarely do we see positive feedback. Okay? All right, so that was our first step. Now, step 2, we're not going to look at in depth because we're not using ATP here. Step 2 is simply taking glucose 6-phosphate and making it into fructose 6-phosphate. Okay? So that's simply this here. That's step 2. But step 3 we're going to look at. And the reason we're going to look at step 3 is because it takes energy. We're putting in yet another ATP. So when we're doing this, this is fructose 6-phosphate into fructose 1,6-bisphosphate. So here, if I'm going to 1,6-bisphosphate, that means I'm adding a phosphate, or I'm phosphorylating that first carbon. Okay, I have to get the phosphate from someplace. Getting it from my ATP again. So my ATP here is hydrolyzed so that I can now have that phosphate to attach to the first carbon. Now, let's look at our enzyme. Our enzyme is PFK1, phosphofructose kinase 1. What did I say is important to note about kinases? And we're going to see this with all the kinases that we have today. They need a divalent cation. Yes, very, very well done. So they need some sort of divalent cation. In most cases, it's actually magnesium, okay? But they do need that divalent cation to be able to do this. And Celestine, love the exclamation point. You're really owning your answers. That's great. Now, once I have fructose 1,6-bisphosphate, okay, we've hydrolyzed our ATP. Again, I used an ATP. This is very exergonic. I have a negative delta G. This is irreversible. Okay, it's not going to be able to go backwards. I want you guys to note this. This is very, very important. This is the rate-limiting enzyme. This is my rate-limiting step in all of the glycolytic pathway. Very, very important. Now, as we look at this, there are always things that can activate, always things can deactivate. If I look at AMP, why would AMP, okay, that's adenosine monophosphate, why would that activate phosphofructose kinase? Why would that do that? Possibly because I used up the ATP, absolutely, because I don't have enough ATP in the cell. And this is telling me we need to make more of this. We need to get into that citric acid cycle. We need to get into the electron transport chain. We need to get ourselves some more ATP. Okay, so if I have AMP, that's going to activate this. ATP, on the other hand, is going to inhibit this. It's going to say, whoa, 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 we got lots and lots of ATP in here, guys. Slow down a little bit. Okay, so that AMP-ATP ratio is really going to determine how well phosphofructose kinase works. That's why this is my rate-limiting step, okay, because it has to do specifically with that AMP-ATP ratio. All right, But what if I still want to make more? Well, in my liver, and this is specifically in the liver, I can use PFK2. It's another kinase. It's going to need a divalent cation. This can actually take and move the phosphate from the first carbon to the second carbon. And I now make fructose 2,6-bisphosphate. If I have fructose 2,6-bisphosphate made by my liver, if I have a lot of it, it can actually activate PFK1. So it can actually overcome if I have inhibition by ATP. So if the ATP ratio is too high, this is a way that I can continue to do this reaction. All right, let's talk a little bit about what happens to the fructose 1,6-bisphosphate. A couple of different things happen to it. It's broken into two 3-carbon molecules, okay? DHAP and glyceraldehyde 3-phosphate. Now, glyceraldehyde 3-phosphate, this can go forward, this can go into our payoff phase. DHAP cannot, however. So I can do two different things with DHAP. I can either use it for alternative pathways, or I can convert it in step 5 into glyceraldehyde 3-phosphate. Okay? All right, couple of great questions coming up about the liver. So first of all, fructose 1,6-bisphosphate this happens in all cells, so it's already going to be in the liver, Kara. Okay? So it'll be taken from those cells that are used in the liver. All right? And this happens if the liver needs to do things. So it's overriding the inhibition in the liver specifically. So the liver is going to be using and needing more energy depending on what it's doing. Okay, depending on if it's converting urea, if it's detoxing cells, whatever that happens to be. So realize that that happens only in the liver. It's not that the liver sends it out to other cells. Okay, so it's the liver that specifically overrides. Does that answer both of those questions? Perfect. Excellent. Okay. So back to our DHAP here. If our DHAP is converted into glyceraldehyde, I now have 2 glyceraldehydes that can go forward. And I really do want to have 2 of them to get to my payoff phase. Okay? So fructose 1,6-bisphosphate is immediately broken down in step 4 into 2 3-carbon molecules. The DHAP can either be converted into the glyceraldehyde 3-phosphate, or it can be used in alternative pathways. All right, so if I have glyceraldehyde 3-phosphate, let's look at what we can do with that. Okay, so my glyceraldehyde 3-phosphate here is going to become 1,3-bisphosphoglycerate. Okay, this is step 6. So we're now working in this direction of our chart. I'm going to flip that over to look at it on the next slide. So we're still going glyceraldehyde into 1,3-bisphosphoglycerate. I want to look at something here. I have an NAD plus going to an NADH. What type of reaction, what is happening to NAD plus when it's gaining that proton? Ah, nice. Very well done. It's being reduced. If it's being reduced, it must oxidize something else. What is it that it's oxidizing? What is it that it's oxidizing here? Yeah, the G3P. Absolutely, the glyceraldehyde 3-phosphate. Perfect. Nice. All right, so always note, if you're making NADH, that NAD plus is helping to oxidize something else because it's being reduced. So it's the oxidizing agent. Now, here we use another enzyme, glyceraldehyde 3-phosphate dehydrogenase, and we get 1,3-bisphosphoglycerate. So now we have phosphates not just on the 3rd carbon, but also back to the 3rd carbon. All right, so we've got 1st and 3rd carbon being phosphorylated here. NADH is generated. Now, when we talk about this, we know that there are 2 NADHs total that are created in glycolysis. That's because from that last step, we have 2 glyceraldehyde 3-phosphates that come in. So each one of those will make 1 NADH. Then we have to think about the fact that this is our high energy intermediate, our 1,3-bis phosphoglycerate. Those phosphates are getting kind of close to each other here. So it's very high energy. This, unlike some of the other steps, is indeed a reversible step. You'll be seeing that when you do Metabolism 2. We will talk about how some of these are reversible and why we would want to reverse them. Now, that's hit that. Now we're going to hit step 7 here. Step 7, this is the first place that I actually make ATP. Okay, this is a good thing. And notice I go from 1,3-bis to 3-bis. So which of my carbons am I going to be pulling that phosphate from to make my ATP? Yep, carbon 1. Perfect. That's exactly what we wanted to look. So step 7 here, 1,3-bis phosphoglycerate. I make ATP. I remove that phosphate from the first carbon, and I get 3-phosphoglycerate. Now, when we look at this, this is done by yet another kinase. Okay, so when we look at this, one of the things that we need to realize is this is what we call a substrate level phosphorylation. So what substrate level means is my ATP is generated, but it's generated without the use of the electron transport chain. Okay, so that's what we mean by substrate level phosphorylation. Anytime we're doing it to a substrate, anytime that we are not using the electron transport chain, this is my substrate level phosphorylation. Okay? And the final thing we need to know about this is, unlike the other kinases, this is actually a reversible reaction. So this is the first kinase that we have seen today that is a reversible reaction. And it's important to know. Our next 2 steps we're not going to look at in detail because we don't make ATP, we don't make NADH. Okay, the next 2 steps, step 8 and step 9. Step 8 simply takes our 3-phosphoglycerate and makes it 2-phosphoglycerate. So all we're doing is moving a phosphate from the 3rd carbon to the 2nd carbon. Then in step 9, we take it and we have our phosphoenol pyruvate. Enol. If I think enol, I should be thinking keto-enol tautomerization. So when we're doing this, this happens because we make that enolate. Okay, so we're going to be going back and forth between the enol and the ketone. Okay, so think about that on this one. And think about which one is more stable using your organic chemistry. All right, this hits my last step. Step 10 of glycolysis. Okay, step 10 of glycolysis. You guys ready for this? We get our final ATP. So when we looked at this, we ended up with 2 glyceraldehydes coming in, which means I make 2 ATP here. I end up with 2 of everything, so I make another 2 ATP here. So what is my net ATP created in glycolysis? My net is 2. What is my gross? What is my gross? Yeah, my gross is 4. So why are you guys telling me that I only made a net of 2? Why are you guys telling me I only made a net of 2? I had those 2 inputs in the beginning. Perfect. You guys got it. All right. Let's look at our final step here. In this final step 10, what we have is our phosphoenol pyruvate. Again, I should be thinking enol, keto. Okay, so I should be thinking about that tautomerization. Here, I make my ATP. So I'm pulling off that phosphate, and I'm making pyruvate. Okay, pyruvate is the last step in glycolysis. When I look at this, this is done by another kinase. I have a divalent cation there. ATP is generated. Again, substrate level phosphorylation, which means that I do not need what to make my phosphate, to make my ATP. Substrate level means I do not need what? The electron transport chain. Exactly, exactly. So Nicole, you are correct in the fact that I don't need oxygen as well, because oxygen is necessary for the electron transport chain. Okay, but the big definition is that it's done without the use of the electron transport chain. Okay, now, fructose 1,6-bisphosphate. This came from step 3, okay? This can actually activate the pyruvate kinase. So what this does is it says, if there's a lot of this around, it says, you know what? We need to activate this. We need to make more pyruvate. This is what we call a feed-forward activation because it comes from an earlier step, but it's allowing a later step to happen. So it's activating it. Final thing is, this is reversible. I can't take pyruvate and go back to phosphoenolpyruvate. Why do I want pyruvate? Why is pyruvate something important to me? Where's that going to go on to? It's used in other pathways. It will eventually get to my Krebs, my citric acid cycle. Now, does pyruvate itself go directly into Krebs? No. No, what actually goes into the Krebs cycle? Acetyl-CoA. Good, Nadine. Take away that question mark. Be bold. Tell me it's acetyl-CoA. So glycolysis happens in my cytoplasm. The step to make pyruvate into acetyl-CoA, first of all, I'm going to ask you 2 questions, so I want to see 2 different things in your answer. Where does that occur, and what is it called? So the process of making pyruvate into acetyl-CoA. Where does that occur? What is it called? Where does that occur, and what is it called? Yes, it can, if necessary, be converted into oxaloacetate. Kara. All right, so what is it called, and where is it located? Pyruvate to acetyl-CoA. See everyone typing. It happens in the mitochondria. Absolutely. It is a type of acylation, Christian, but there's a specific step in our cellular respiration. Okay, so it definitely happens in the mitochondria, but there's a specific step. It's got a bigger name than that. So we have glycolysis. We could have fermentation if we didn't have oxygen, but instead we go into which step? Okay, does everyone remember the name pyruvate decarboxylation? Yeah, long name, yes, pyruvate decarboxylation. So we could take this pyruvate and go into fermentation. And with that, we'd make 2 lactic acids. That's not our norm. That's only when we're in an oxygen-deprived state. So they're not going to really ask us about the steps of fermentation. We need to understand that fermentation occurs because of oxygen death. Okay? But we do need to know that the pyruvate actually goes into the mitochondria and goes through pyruvate decarboxylation. Okay? Notice that pyruvate decarboxylation is not one of our goals today. Our goals are glycolysis enzymes and citric acid cycle enzymes. The reason for that is they're really not going to ask us about the enzymes in pyruvate decarboxylation because there's only one. Okay, it's called pyruvate decarboxylase, and that's what's going to get us to our acetyl-CoA. Okay? All right, so let's look at the overall, what we talked about, our overall 10 steps here. Important to remember that hexokinases are going to use magnesium, okay, or another divalent cation. These are all going to use magnesium. Then, as we continue to go through, we used 2 ATPs. In this step, we made 2 glyceraldehydes, so we're going to get 4 ATPs out total. Also remember, step 3 here with my PFK1 is my rate-limiting step. That can be overcome in which organ? I can overcome the inhibition of PFK1 in which organ? Yes, yes, my liver is special. I love it. It's my friend. Okay? All right, so the net out of this, we get 2 NADH total. That comes from our 2 glyceraldehydes. We get 4 ATP total, but we use 2, so it's a net of 2. And then we get 2 pyruvates. These 2 pyruvates are going to move on. All right, let's ask ourselves a quick question about our glycolysis. Which of the following, if its cytosolic concentration were increased, would have the least effect on the activity of phosphofructose kinase? Okay, so this is talking about PFK1. What happens in PFK1? What happens in PFK1? ADP is made. ATP goes to ADP. All right, so which would have the least effect? Okay, so 3 of these will definitely have an effect, 1 will not. And what affects that? Go back and look at that slide. What affects that? It's a specific ratio. What ratio? Yeah, the AMP ATP. Okay, and AMP goes to ADP before it comes ATP. So it's mono, di, and tri. So all 3 of those would absolutely affect the PFK. Now, that means that the one that wouldn't is inorganic phosphate. Why would inorganic phosphate, having phosphates running around, why would that not affect this? Excuse me. Why would that not affect this? Let's get our critical thinking skills going in here. Thanks, Kyle. Yeah, so it's removing a phosphate. I'm going ATP to ADP. So if I have extra phosphates around, is that going to change anything? No. I'm still going to be going ADP or ATP to ADP, and that phosphate will be transferred to fructose 1,6-bisphosphate. So having more hanging out in the cell isn't going to do a thing. All right, great. Here's a great question that they could potentially ask us. This is letting us know what is reversible, what is not reversible. Which of the following enzymes catalyzes a reversible reaction in glycolysis? So I look at these and I have to think about these. Hexokinase, that was our very first one. We said that is absolutely irreversible. Phosphofructose kinase, we said was absolutely irreversible. And pyruvate kinase, making that pyruvate molecule, we said is absolutely irreversible. Well, the only one we didn't list in that is the phosphoglycerate kinase. That one actually is reversible. All right. That's Learn Go 1. That is our glycolysis. Let's now talk about the citric acid cycle. We love the citric acid cycle. This is where we start seeing the payoff. So the first thing that happens here is our pyruvate decarboxylation. When we do our pyruvate decarboxylation, what we see is one enzyme. Okay, so here's our one enzyme. Pyruvate going to acetyl-CoA. Question for you. NAD here is being reduced, which means what's happening to my pyruvate? What's happening to my pyruvate? It's being oxidized. Now, specifically, it is the carboxylic acid on my pyruvate that is being oxidized into carbon dioxide. Okay? So, this is going to be specifically the carboxylic acid into carbon dioxide. All right, here, this is important. This is huge complex here. So this is a huge, huge complex that not only has magnesium, but there are other enzymes involved in this. So this is a 5-enzyme complex called pyruvate dehydrogenase complex, or PDC. This is going to get us from our 3-carbon pyruvate into our 2-carbon acetyl-CoA because we're losing 1 carbon dioxide. We know this occurs in the mitochondrial matrix. We know that it generates an NADH, and it produces acetyl-CoA, which is my main substrate of the citric acid cycle. Now, let's look at what can cause it to occur or what can inhibit it. Anytime I have a lot of NAD+, anytime I have a lot of ADP, this is telling me I need to use these. I need to make more ATP. That's going to activate this. Calcium will also activate this. Calcium is actually the divalent cation that we would be using in our muscles specifically. Then to turn this off, if I have lots of NADH, if I have lots of ATP, or if I have lots and lots of acetyl-CoA, that's going to inhibit this complex, this 5 enzyme complex from working. All right, so other things can happen if we need acetyl-CoA. We can use things like fatty acids, ketone bodies, alcohol to get this. And this is something that you will talk about in Metabolism 3. All right, so let's look at our citric acid cycle. I made my acetyl-CoA. Woohoo! That's what goes in here. So we're going to talk about our very first enzyme, which happens to be citrate synthase. So this here we're making citrate. So we take our acetyl-CoA and the oxaloacetate. So if we go back here, notice that the oxaloacetate and the acetyl-CoA are going to come together to make my citrate. When I do this, I have water going in, okay? I make my CoA come back out. And here I use citrate synthase. Citrate synthase. Now, let's look at what we have. Acetyl-CoA is a 2-carbon. Oxaloacetate is 4-carbon. And that means that citrate is going to be how many carbons? Because all that's coming off here is my coenzyme A. Yeah, that puppy's going to be 6 carbons. Excellent. Good. This occurs in the mitochondrial matrix. Okay, this is something that is a condensation reaction. We see that with the water. And this is irreversible. Okay, once I make my citrate, it cannot go backwards. Now, I again can activate and deactivate. Anytime I have NAD or ADP, this tells me I need more NADH, I need more ATP, that's going to activate. Anytime I have my NADH, my ATP, or succinyl-CoA, this is going to come from a future step. So we're going to hold on to this in just a minute, but this comes from a future step. That can also deactivate this because that is a downstream enzyme or a downstream substrate that comes later in the citric acid cycle. So if there's too much of it, I will come back here and I will inhibit citrate synthase. Citrate, because it's the product, will also be something that inhibits this. That doesn't surprise us. Products can typically inhibit our enzymes. All right, that was our step there. Let's now look at step 2. Citrate to isocitrate. Citrate to isocitrate. Here I have water, and this can go in either direction. I want you to notice there's an intermediary here. Okay, cis, which means it was added to the same side. Cis, I can never pronounce that, aconitrate, okay, or nitrate. This is our intermediate. Here's the deal. We use the same exact enzyme for both of these reactions, and it's reversible here. Okay, so we use the same exact one. Notice I have another divalent cation. This one's a little bit different, but I still have a divalent cation on here. So this is aconitase, and this is going to do both of these reactions. They are indeed reversible. Okay, so what I'm doing here is I'm actually making an isomer. Okay, so this is an isomerization reaction. If we look at citrate, we have a tertiary alcohol. If we look at isocitrate, what type of alcohol do I have here? What type of alcohol do I have? Secondary, good. So if I'm looking at alcohols, the whole idea behind this is to make a form of alcohol that is much more easy to react. So I'm going from a tertiary alcohol to a secondary alcohol. That's the whole idea behind this, okay, is to create that alcohol that is much more easily oxidized. All right, I have isocitrate lyase, or isocitrate. So isocitrate will then turn into alpha-ketoglutarate. Now, some of us probably have heard of the isocitrate lyase shunt and the malate synthase shunt. Notice that those are not on this picture. So you don't need to know those two shunts in here. Okay? All right, so isocitrate into alpha-ketoglutarate. And yes, Christopher, the same enzyme performs that reaction, both directions, and it's in 2 steps. Absolutely. Okay? All right, so our isocitrate here, I have to take that to alpha-ketoglutarate. NADH, or NAD plus, is being reduced to NADH, which means my isocitrate is being oxidized. Okay? So, as I do this, I'm going to have, again, an intermediate here, and then I'm going to get my final piece. So, my IDH, another thing that needs a divalent cation, isocitrate dehydrogenase, is going to take my isocitrate, dehydrogenate it, well, that makes sense, I see a proton here, into oxalosuccinate. Then IDH, again, the same enzyme, is going to take that proton and put it on my alpha-ketoglutarate and remove a carbon. So isocitrate was a 6-carbon sugar. How many carbons are in my alpha-ketoglutarate? 5, because I just lost a carbon dioxide. The only place that that can come from is my 6 carbons. Perfect. All right, again, mitochondrial matrix. This is our very first step in the citric acid cycle that creates NADH. Okay, so when we say first step to generate it, we mean specifically in the citric acid cycle. And then this is the first one to produce our carbon dioxide. As we look at that, what happens to the carbon dioxide? What happens to that after I make it? Where does it finally end up? Where does it finally end up? Yeah, it's exhaled. Good, Sarah. It leaves. It binds to carbonic anhydrase. Yes. Makes carbonic acid. When it gets back to my lungs, converts back to carbon dioxide, and I exhale it. Perfect. Notice the arrows, unlike the last one, are not going both directions. So this is an irreversible reaction. Let's look at what can activate and deactivate IDH. Having a high amount of NAD+, having a high amount of ADP, this tells me that I need to be making NADH and ATP. That will activate, and calcium will activate specifically in my muscle cells. If I have a high amount of NADH or ATP, that will inhibit IDH. All right, now we've got our alpha-ketoglutarate. We have to turn this into succinyl-CoA. Succinyl-CoA we've already seen. It actually inhibits the step that's going to isocitrate. All right, so now when we look at this, this is our step 4. Our alpha-ketoglutarate, notice I have a coenzyme coming in here, and I get succinyl-CoA. So anytime a coenzyme comes in, I get something CoA. Anytime it goes out, that goes away. I have another reduction of NAD, so that means I have another oxidation of my substrate. With that oxidation, again, I'm going to be creating a carbon dioxide. So I'm going to be taking a carboxylic acid, and I'm going to be creating a carbon dioxide. I have another multiple enzyme complex here, alpha-ketoglutarate dehydrogenase, and I'm going to get my carbons out. So I went from a 5-carbon to a 4-carbon, okay, because I just lost another carbon dioxide, which I will exhale. This occurs in the mitochondrial matrix. It is irreversible, and it's the second step in our citric acid, yes, to produce NADH, which we need, and our carbon dioxide, which we will exhale. To activate this, again, notice the pattern here. All of these are being activated by a high amount of NAD+ or a high amount of ADP. Okay, so these are the substrates that are used to make our NADH and our ATP. So when we look at this, this is a lot of the same thing going on. And again, in our muscles, calcium will cause this to work as well. High amounts of NADH or ATP or the product will turn this off. Again, are you guys noticing a pattern here about what is actually inhibiting these different enzymes and what is actually activating these three enzymes? Nicole, they have actually asked us, yes. They have stated that you need to know different parts of citric acid cycle. You need to be able to list these things. Absolutely. Okay. There can be some exceptions to those, okay, Christopher, but in general, anytime you have an enzyme, in general, you are going to see that the product is going to come back and inhibit it, okay, which is good. This is why we use enzymes. All right, now, the other thing that succinyl-CoA will inhibit, remember way back in the beginning when we were making our citrate, the citrate synthase will be inhibited by succinyl-CoA. So the making of acetyl-CoA into citrate can also be inhibited by this. This is just a downstream product, but it's the same idea. It's still a negative feedback. Okay. All right. Sarah, Mike has got that for you. Okay, Mike has got that for you. All right. Now, we've got our succinyl-CoA. So we're down to our next piece, which is making our succinate. Succinate, this is our fifth step here. And succinate, when we look at this, we've got our CoA and succinate. Notice the CoA went away, so my coenzyme is coming off. These coenzymes are rejuvenated. These coenzymes are continually made. What do you notice about this step that is different in here when we're looking at what goes in and comes out from any other step that we've ever seen to this point? Ah, yes, I'm making GDP, not ATP. This is the only step in which I make GDP, okay, GTP. So this happens with succinyl-CoA synthetase, okay, so that means we're synthesizing something. This occurs in the mitochondrial matrix, substrate level phosphorylation. Okay, substrate level phosphorylation means I don't need electron transport chain. As I look at this, this is the only one that makes GTP, and the GTP will immediately convert into ATP. Yeah, exactly, Nadine. It'll immediately convert over. So we can use GTP in our bodies, but it's much more difficult to use. So this will immediately be converted into ATP. All right. Now, what can inhibit this? What can keep this going? There's not big inhibitors there, so that's why we don't look at them. Okay. Our next step, step 6, is looking at succinate into fumarate. Okay. Now, succinate into fumarate is FAD going to FADH2. What is happening to my FAD? While you guys are typing in what's happening to my FAD, Kara's got a great question. GTP is produced instead of directly producing ATP. You have to remember that our mitochondria came from aerobic bacteria. So though it's hard for us to use, it was a thing that was created long, long before we were. Okay, so we just have trouble using it. Does that make sense? Okay, perfect. All right, so succinate, if my FAD is being reduced, my succinate is being oxidized. Now, there's something special about this enzyme, okay? This enzyme is the only enzyme in looking at our citric acid cycle that is actually bound, okay? And this is bound into the membrane of the inner mitochondrial membrane. So this is actually a membrane protein of the electron transport chain. Okay, so this one is bound for the electron transport chain. Okay, now occurs in the inner mitochondrial membrane because it is a membrane-bound enzyme. So this is actually part of the electron transport chain. FAD is covalently bound. Okay, so it's covalently bound, which means it doesn't go anywhere. That's why this has to be part of the electron transport chain, because the FAD we make, the FADH2 we make, each of those will make 2 ATPs. So this has to be bound into the membrane because it's actually part of the electron transport chain. Okay? Then, when we look at this, it's the only step ever that produces FADH2. Okay? And remember, as we're talking about the citric acid cycle, this is one turn, so one acetyl-CoA, but we're going to have 2 acetyl-CoAs that come in. So everything that we do happens twice from every glucose. All right, we're getting down to the end here. In step 7, malate is formed from fumarate. Okay? Now, malate is formed from fumarate, and I know we skipped a step here. We just ended with succinate and going into fumarate. Okay? So fumarate goes into malate in step 7. And when I look at this, I've got water coming in. Okay, if water's coming in, there's actually a double bond in fumarate, and water is going to be added across this double bond. This is done by a simple little enzyme called the fumarase enzyme. Nothing else in here this will always happen. Nothing really inhibits this one. And then our final step here, okay, this occurs in our mitochondrial matrix. Our final step is going to be malate to oxaloacetate. So notice, remember at the very beginning, acetyl-CoA and oxaloacetate had to go together to make citrate? We're actually rejuvenating the oxaloacetate. So that, as this continues to happen, that's going to continue to be rejuvenated so it can continue to be used, which is really, really nice. Okay, so this is reformed. When we look at this, we have our final reduction, which means malate is going to be oxidized. This happens with malate dehydrogenase, and we finally end up with our oxaloacetate going back to the beginning. Okay, so as we look at this, this occurs in the mitochondrial matrix. This occurs and oxaloacetate is regenerated, and this is our final step. Now, they could ask us a simple question like, which is the only enzyme in the citric acid cycle that does not occur in the mitochondrial matrix? What would your answer be to that? What is the only enzyme that does not occur in the mitochondrial matrix because it's bound? Yes, SDH, succinyl dehydrogenase, because it's bound as part of the electron transport chain. Nice. Only one. So knowing those odd guys out can really help you here. Okay, so let's go back and let's talk about what would inhibit and what would activate this. So if I have ATP and NADH, this inhibits several steps in here. This says I have enough ATP, I have enough NADH. This is going to inhibit PDC. Notice NADH is being made there. This is going to inhibit CS, where I make my citrate. This is going to inhibit IDH. Notice we make NADH there. And this is going to inhibit alpha D KDC. So when I look at these, the only one that does not produce NADH is going to be CS. Okay? It also will not inhibit oxaloacetate. Okay, that last step, that's not going to be inhibited. But every other place where I make NADH, this will be inhibited. If I have ADP and NAD+, I'm energy deficient, what am I going to activate? Well, this is easy. If I remember the 4 enzymes that ATP and NADH inhibit, ADP and NADH+ will activate. So this is my PDC, my CS, my IDH, and my alpha KDC. Okay, so notice, yes, and I like that, the steps that make the NADH. Exactly, that's when we're energy deficient. Perfect. Okay, here's our summary. Now, there are a couple of things that inhibit other things. So if I look at succinyl-CoA down here at the bottom, which earlier enzyme does that inhibit? Which earlier enzyme does that inhibit? CS. Yes. Good. So acetyl-CoA can inhibit the enzyme that makes it. Citrate can inhibit the enzyme that makes it. Succinyl-CoA can inhibit the enzyme that makes it, or CS. Now, even though these are inhibited, in our muscle cells, we can use which divalent cation to activate these? Yes, good, Christopher. Calcium. Calcium, because my muscle cells need a lot more energy than some of the other types of cells for my body. So calcium in my muscle cells can activate PDC, can also activate IDH and alpha KDC. The only one that it does not activate is our citrate synthase. All right. Yeah, and it causes muscle contraction. Absolutely. So let's look at our net. First, we're going to look at pyruvate decarboxylation. Here we get 2 pyruvates used, we get 2 acetyl-CoAs made, and we get 2 NADHs made. That's our net for pyruvate decarboxylation in the mitochondrial matrix. Then in the mitochondrial matrix with my citric acid cycle, I get 2 acetyl-CoAs, 6 NADHs, 2 FADH2s, and 2 GTPs, which will be converted to ATP. So all of this comes from just one glucose. And again, FADH2 is actually part of the electron transport chain, so SDH is in that inner mitochondrial membrane. Know what is net made here. All right. Whew. It's a lot of information. Okay, but we absolutely have to know this for test day. The AAMC has said on the new MCAT, this is absolutely testable material. All right, let's look at one question. Okay, we're going to look at one question together. We're almost done. I know, Nadine, it's kind of a bummer, but we do have to know it. Okay? In this case, yes, I do agree. Okay, but this is more critical thinking. If you know, they will probably ask you about one step, be honestly, and if you know that, for instance, you know, you're going from fumarate to malate, it's going to have a fumarase in there. Okay. All right, let's look at this one question, and then we'll look at our review. In plants, the glyoxylate cycle converts acetyl-CoA for use in gluconeogenesis in a special organelle called the glyoxysome. If the glyoxysome lacked, it's always the one that I cannot pronounce, aconitrase, the synthesis of which of the following intermediates found in both the glyoxylome and the citric acid cycle would have to take place in the cytosol. So what does ACO make? Aconitrase. What does that make? Yes, it makes isocitrate. So it's citrate to isocitrate. So which would have to take place in the cytosol? I know what that is. That's isocitrate. We know that. How do we know that, Nadine? Because that is the second step in the citric acid cycle. Okay, so we know that because that's the second step in the citric acid cycle. All right. All we have left. I want you guys to do this one at home as homework. Again, the benefit of homework is being able to look at some of the information that we went through and doing it on your own. So metabolism 1, we looked at glycolysis. This converts glucose into 2 molecules of pyruvate. There are irreversible enzymes of hexokinase, phosphofructose kinase, and pyruvate kinase. This is inhibited if we have lots and lots of ATP. It's stimulated by a high amount of ADP. The citric acid cycle uses the acetyl-CoA that comes from pyruvate decarboxylation to produce electron carriers of NADH and FADH2 for the electron transport chain. There are three different enzymes that are irreversible. That's citrate, synthase, IDH, and alpha KDH. It's inhibited by high amounts of ATP and NADH, and it's activated by low amounts or high amounts of ADP and NAD+. All right, I know this is a lot of information. Congratulations on getting it through it, and we will see you tomorrow or in another future channel session. Have a great day. Bye.